Good afternoon, ladies and gentlemen. Thank you for the introduction. I will give you a brief overview today about mutation profiling of biliary tract carcinoma, a systematic review and meta-analysis. I have nothing to disclose. As we all know, you have different forms of biliary tract carcinomas. And as Dr. Primrose already stated yesterday, you have lumpers and you have dividers. And as we know, in surgery, we have an anatomical divide. We have intrahepatic cholangiocarcinomas, gallbladder carcinomas, extrahepatic cholangiocarcinomas that are further subdivided in perihilar and distal cholangiocarcinomas. However, biologically, these tumors are related, and this has mainly to do with the fact that the cell of origin always derives from the biliary tract epithelium. Either they are dedifferentiated mature cells, or they are cells that derive from the canals of Herring in the small bile ducts that arise into intrahepatic cholangiocarcinomas, or they derive from the peribiliary glands, and these are lying in the larger bile ducts. And as a matter of fact, this whole tract is only 7 centimeters long, so you can imagine these tumors are related. And as we do molecular research in our department, we are actually quite curious in how often these tumors were related and in which way mutations were shared or were separate. And therefore, we did a systematic review and a meta-analysis. We included all studies that reported on next-generation sequencing on biliary tract carcinoma from, the point that sequencing started until the end of 2017. We included 25 studies with 1,806 patients, of whom the majority had intrahepatic cholangiocarcinoma. And all studies had reported on whole exome, whole genome, or target sequencing. And as we move on to the shared mutation, the interesting thing is that most biliary tract carcinomas actually share a common blueprint in mutations. They all have TP53 mutations, ARID1A, MUC16, KRAS, and SINA1 mutations. But they all range from 10 to 30% of patients who are affected with these genetic alterations. Interestingly, there are many intrahepatic cholangiocarcinomas and extrahepatic carcinomas that are quite related. They share some mutations in SMET4, SEX, and BAP1, and these are also mutations we know from pancreatic carcinoma. Interestingly, gallbladder carcinoma looks quite different, especially from intrahepatic carcinomas. And extrahepatic and gallbladder carcinomas also share more mutations than the previous two. If we look at exclusive mutations, which is something we are always interested in, you see that there is a subtype of intrahepatic cholangiocarcinomas that exhibit IDH1 or IDH2 mutations and also FGFR2 fusion genes with different fusion partners. And in extrahepatic cholangiocarcinoma, you see that those tumors are more related to gastrointestinal tumors and pancreatic tumors. And they have mutations in APC, GNAS, and TGFB2 genes that we also know from, for example, colorectal carcinoma and periampular carcinomas that have intestinal subtype. Gallbladder carcinomas are quite distinct, and they have mutations that involve the cell metabolism. So in conclusion, what we see is that all types of biliary tract carcinoma share a specific blueprint, but also have some very exclusive mutations that are probably related to their cell of origin. And this heterogeneity is interesting because we furthermore see that the frequency of mutated genes is also quite low, around 30% maximum, while in pancreatic carcinoma, for example, 80% of those carcinomas have a KRAS mutation. This might be due because there are lots of epigenetic differences on which I will not further go into, but it's actually quite interesting. And what do we do with this information? Well, for the understanding of these tumors, this is very important because otherwise, when we don't understand genomics, how can we understand the biology of these tumors? There are some new molecular diagnostic purposes, and there are also, even now, more targeted therapies, even for cholangiocarcinoma. So it is interesting to know which mutations affect which subtypes. And therefore, I cannot emphasize that more. It is very important that people share their genetic and genomics data on these rare diseases, not only in their papers, but also on the platforms that are developed for this. Thank you for your attention.
Any questions or comments?
I think not.
I think we can close the session. Thank you very much for all the presentation, and I hope you enjoyed it. Have a nice evening. Bye.
Thank you.